Hello, my name is Kea Schwarz, and in my presentation we will discuss gut hormones and satiety. The World Health Organization has listed obesity as an epidemic. It's not obesity itself, but the many diseases that are associated with obesity like diabetes, hypertension, heart disease, and cancer. Medical treatment of obesity is relatively ineffective. Under a program of healthy eating, exercise, drug therapy, psychological intervention, a weight loss of up to 20 pounds can generally be achieved, but long-term application of any program is rarely achieved. This is why in 1991 the NIH endorsed bariatric surgery for the morbidly obese. Bariatric surgery offers a surgeon the opportunity to cure diseases and prevent further diseases. What I mainly want to talk about today is the hormones that are responsible for promoting satiety, such as insulin, leptin, cholecystokinin, peptide YY, glucagon-like peptide, and pancreatic polypeptide. We will also talk about ghrelin, the hunger hormone. We will review the pathway of these hormones as well as look at how satiety hormones are influenced after bariatric surgery. All of the studies reviewed looked at the effect of bariatric surgery on gut hormones and their relation to weight loss. Roux and colleagues examined meal-stimulated responses of insulin, ghrelin, peptide YY, glucagon-like peptide 1, and pancreatic polypeptide after bariatric surgery. Corner and colleagues looked at 4 groups of women: a lean group, one group that went through gastric banding surgery, another group that went through Roux-en-Y gastric bypass, and a control group. In both Roux and Corner's research, there were similar results. The Roux-en-Y gastric bypass group had an increased postprandial plasma polypeptide YY and glucagon-like peptide 1, favoring enhanced satiety. Also, the Roux-en-Y gastric bypass patients showed exaggerated insulin responses, potentially mediating improved glycemic control. The Roux-en-Y gastric bypass groups also showed an overall greater level of satiety over the other bariatric surgeries performed. Additionally, leptin and ghrelin levels were similar between the 2 surgical groups. Another study performed by Cummings and colleagues looked at the 24-hour plasma ghrelin profiles among other satiety markers in 13 obese subjects before and after a 6-month dietary program for weight loss, also in 5 patients who had lost weight after a gastric bypass, and 10 normal weight controls. 5 of the 13 obese subjects who participated in the dietary program were matched to the subjects in the gastric bypass group and served as obese controls. The results showed a lower level of ghrelin in the weight loss group that had gastric bypass surgery as compared to the control group that lost a lower amount of weight through diet. This suggests that gastric bypass aids satiety even after extensive weight loss. Similarly, Corner and Roux also found low levels of ghrelin in all surgical groups that had undergone gastric bypass surgery. Insulin, leptin, and ghrelin are some of the main players that determine hunger and satiety. These hormones communicate with the hypothalamus to help us determine if we need to intake more energy in the form of food. Eating slowly fills the stomach, which causes the stomach to stretch. Nerves in the stomach sense the stretching and send a signal via the vagus nerve to the brainstem and the hypothalamus. These are the main parts of the brain that control food intake, but this is just one way the body recognizes satiety. It also takes into consideration hormones, which are produced from the endocrine cells that are found throughout the digestive system. These respond to specific nutrients like fatty acids, carbohydrates, and amino acids in the gut and bloodstream. Some of the hormones that are released from the intestines in response to food intake are cholecystokinin, peptide YY, pancreatic polypeptide, and glucagon-like peptide 1. These are all responsible for promoting satiety. After a meal, the amount of glucose in the blood increases. This triggers the release of insulin from the pancreas. Insulin helps control blood glucose levels by signaling the liver and muscles to take in the glucose from the blood. Insulin therefore helps cells to take in glucose to be used for energy. If the body has sufficient energy, insulin signals the liver and muscles to take up the glucose and store it as glycogen. Insulin also stimulates the release of leptin from adipose cells. These hormones travel to the hypothalamus and inhibit receptors on neuron populations to suppress hunger. A few hours after a meal, our stomach will be empty and it will release another hormone called ghrelin into the bloodstream, which will travel to the hypothalamus to increase the activity of hunger causing nerve cells, which lets the brain know that you're hungry. This will motivate you to find food and starts the cycle all over again. Under normal conditions, the body is equipped with proper cues to help you determine when you are hungry and when you are satisfied. These hunger cues can fall out of balance for many different reasons and can lead to obesity. Overall, bariatric surgery is the most effective therapy for severe obesity in terms of weight loss. These studies presented all show an increased concentration of the satiety hormones such as polypeptide YY and glucagon-like peptide 1 after bariatric surgery. Ghrelin levels were also shown to decrease to a lower amount when compared to control groups. There were slight differences in hormone levels based on the type of bariatric surgery. Roux-en-Y gastric bypass seems to be the most successful in promoting satiety and increasing weight loss. That being said, understanding how these hormones work in the body under normal and adverse conditions like obesity helps dietitians determine the best course of action when delivering a meal plan to an individual in different levels of health and fitness.